All right, my friends, are you sick and tired of chapter 12 yet, or what? Yes. It's like we're going there forever. Today, we'll be done with chapter 12. Yay! All right, so we left off talking about cerebrospinal fluid, which is for what? Nourishment, number one. Get rid of waste. Protection, cushion. When we talk about fluids in any body compartment, that's one of the things we're going to always mention, the fact that fluid helps to cushion and protect. The volume of cerebrospinal fluid is also very important because that sets up a pressure gradient. And anytime I set up a pressure gradient, it's important that I stick to that gradient. Things want to go from a high pressure to a low pressure. And in order to do that, we have to have that pressure gradient set up. If I start messing with the pressure and creating too high a pressure, I'm not going to have transfer of materials in the proper way. Also, too much pressure from cerebrospinal fluid can cause a lot of problems with the neurons and neuron function itself, and can actually cause damage. So, helps to give buoyancy to the central nervous system, which protects it from trauma and blow and provides nutrients. When we look at flow of cerebrospinal fluid, this diagram on page 461 is a nice one showing us where fluid is produced. And where is cerebrospinal fluid produced? The ependymal cells of the ventricles, specifically.
Ken, what is it?
There you go, choroid plexuses. Very good. So in this diagram, it's these little red regions we see in the brain ventricles. And this diagram just shows us the flow of cerebrospinal fluid through the central nervous system. Remember, it flows down the spinal cord too. What's that big hole down the central of the, spinal cord? Central canal, and that fourth ventricle is going to connect to the central canal to provide flow of cerebrospinal fluid through the central nervous system. So the choroid plexuses are places where we're going to actually produce this stuff and reabsorb this stuff in areas called the arachnoid villi. So we produce it at a constant rate and we keep it in motion. This allows for efficient nutrient delivery. Very vascular in those areas as well. Makes sense, right? Because where are we getting the materials to make our cerebrospinal fluid from? The circulatory system. So it's what we call an ultrafiltrate of blood plasma. If we have to get rid of waste, where are we going to get rid of it too? Circulatory system as well. So we use ion pump controls to make sure that we have the right materials for cerebrospinal fluid, and we also get rid of cerebrospinal fluid, older stuff, at the proper rate. About 150 milliliters, and we replace it every 8 hours. So we're constantly making new. This is just a diagram that shows us up close and personal those ependymal cells and the circulatory system vessels associated with the choroid plexus regions. That's why they're red on the diagram, because they're highly what? They're highly vascular. So if too much pressure builds up in the central nervous system, we can have lots of problems. If there's some sort of obstruction in the reabsorption process in those arachnoid villi, improper drainage, too much production, we can cause some real problems in the central nervous system. Another thing that can be a problem is when infants are born with fused skull bones. What's there before they become fused? What's there before sutures form? It's cartilage. What do we call them? Fontanelles. And that allows for the little bit of expansion that has to take place before those bones actually form the sutures. If children are born with these bones already fused, and that happens, that can be a real problem with pressure buildup in the central nervous system. So it can result in brain damage. Shunts can be put in to help drain excess fluid to relieve that pressure. On page 462 in your textbook, we see this picture of a hydrocephalic infant. So we have to remedy that ASAP, or there's going to be permanent and major damage to the central nervous system in this child. Those ependymal cells create something called the blood-brain barrier. I don't know why I can't say that. Blood-brain barrier. And this provides a stable environment in the central nervous system by providing stable nutrients for those cells. These cells are very particular as to what they're going to absorb from blood plasma. These are the reason why drug companies hate making drugs that are going to affect the central nervous system. Because, yeah, they might come up with an awesome pharmaceutical that would do whatever it is they desire. But if they can't breach the blood-brain barrier, if the ependymal cells won't carry that substance into the central nervous system, it's not going to do you any good. So it creates, or helps to create, it's true, and so many, and you know, people talk about the cost of drugs these days, but the cost of the research that goes into making those drugs is crazy. And you can be working on something for many, many years and then get to the end stage and it's no good. You have to scrap it and start from the beginning. So you've spent thousands and thousands of dollars for nothing. So it's a tough business. But the blood-brain barrier is going to allow for a continuous composition in cerebrospinal fluid. We see that they line the endothelial capillary walls, those ependymal cells, and thick basal lamina around capillaries. What's a basal lamina? Basal lamina. The basal surface of, yeah, the bottom of those cells in that connective tissue region where the capillaries are. That's basically what that means. The other cells that are going to help these cells create efficient spinal fluid is the astrocytes. Remember those guys? Yeah, the most abundance of the glial cells in the central nervous system. But those legs are going to help create a connection between the ependymal cells and neurons and capillaries and neurons to help get nutrients to the neurons when needed. So that barrier is very selective, only going to allow certain things to pass through, making sure we get rid of metabolic wastes, drugs, non-essential amino acids, potassium, all of these things. Nope, we're not going to let in. Some things make it pass, though. Allows any fat-soluble substance to pass, including alcohol, nicotine, and anesthetics. So if I'm a drug developer and I'm going to create a drug, what kind of organic molecule am I going to try to work towards? Yeah, some sort of lipid-based carrier. So, absent in some areas, example, the vomiting center and the hypothalamus, where necessary to monitor chemical composition of blood is needed to help maintain homeostasis. So we have to know what's going on in the circulatory system in order to send messages to these, or send messages to the centers that are being sent. Wherever the hypothalamus sends messages to, we need to have a true value of what's in plasma. So we don't want to expose those cells to something that's an ultrafiltrate of plasma. Does that make sense? No. So no. Okay, let me say it a different way. Those neurons have to know what the true composition of plasma is. Does that make sense? Because they're monitoring what's going on in blood plasma. The reason they're monitoring is because they have to then send messages to areas to help maintain homeostasis. So they have to send messages, for example, when we talk about the hypothalamus, we talk about it all the time. It's a busy, busy little portion of the midbrain. But if I have to know what's going on in blood plasma so I can signal the pituitary gland, for example, to secrete hormones, if I'm exposed to an ultrafiltrate of plasma, I'm not going to get the true picture of what's going on in the body. Does that make sense?
How is the hypothalamus protecting it? If there's filtrates and toxins in the blood, is there any kind of protective layer for the...
No. So you have to, that's why you have to keep in mind that that really important part of the brain is going to be exposed to whatever you put in your body. And when they talk about things like certain drugs, for example, and losing neurons, we're talking about losing neurons in an extremely vital control center in your central brain region that controls so many different things, as you well know from studying chapter 12. So those are the kind of neurons you're going to lose first. Do you want to do that? I say no. Not worth it. Homeostatic imbalances of the brain, talked about next on page 462. Different things can cause problems with neuron function and actually alter message being sent in and out. Concussion, what's that? Anybody watch football this weekend?
Yeah, that's when the brain...
You bang it so hard, it... Exactly. So you're going to get like a bruise on your brain matter when you smack it into the side of your skull. Contusion can be permanent damage. Subdural or subarachnoid hemorrhage. Subdural under the what? Dura mater, or subarachnoid under the arachnoid mater. Remember, we had spaces there. And in those spaces, not only are we going to find cerebrospinal fluid, but we're going to find blood vessels as well. Anytime I don't get nutrients to an area, and the guys carrying most of the nutrients are going to be who? Circulatory system, yes? It's going to result in starvation in certain areas. So hemorrhage to a region might result in starvation of cells in those regions, and that can result in death of those areas. And depending on where those areas are, it can result in death, period. Cerebral edema,
It's terrible.
Yeah.
A friend of mine was playing darts one day. I mean, he loved to do it, but he stepped up to the line and went to shoot a dart. Fell on the ground. He was dead. And he
Yeah.
just danced to doing it.
Yep. And unfortunately, it was one of those hemorrhages. Yep. That's scary. Scary. Also edema, pressure. When we build up pressure inside the central nervous system for whatever reason, swelling to the brain for whatever reason, trauma can cause increase in pressure in the system. When we increase pressure in the system, we're going to start damaging neurons and neuronal pathways. Cerebrovascular accident, CVA, or stroke. You've heard of that. Ischemia, when I deprive tissue of blood supply. That can be a problem for whatever reason. Usually some sort of blockage is the culprit. Paralysis on one side. Hemiplegia. Transient ischemic attacks. These are sort of leading up to the big blockage, and there's temporary blockage. Many times people who have had a stroke find out that they've had these transient ischemic attacks prior to their major stroke. One of the things that's going to help a patient who is suspected of having a stroke is something that can dissolve those blockages. Yep, and that's called tissue plasminogen activator. When we talk about blood and accessory, when we talk about the clotting cascade, this particular activator is going to disrupt that clotting cascade and help dissolve any clots that might be forming, resulting in a stroke. So if you can get that patient in there as soon as possible and administer this plasminogen activator, there's a good chance that that clot won't cause permanent damage.
What's the drug that does that?
This.
Beta?
No, that's heart. Beta blockers.
That's something that helps with
Anticoagulants, but this is like anticoagulants on steroids.
so this is
So when,
extreme level.
yeah, it's going to interrupt the cascade and actually cause dissolution of clots that have started to form. You're thinking of heparin or Coumadin.
Right.
Those are anticoagulant drugs that help. This is a little bit more active in dissolving clots that may have started to form. Those guys are going to prevent clots from forming. Different disorders, and what I want you to know from this information is a basic definition of some of the disorders described. Alzheimer's disease, problems starting with short-term memory, then leading to long-term memory. And again, think of all those file cabinets you have in the cerebral cortex. When you no longer have the ability to access information from those file cabinets, you no longer have the ability to perform day-to-day functions.
So the brain starts to degenerate.
Yeah, and when the pathways to that information start to degenerate, the information is still there, but the pathway degeneration is what the problem is.
Folks look to
Well, if you don't use it, you lose it. You ever hear that? Exactly. So you start to see,
and the sulcus still turning.
yeah, and you'll start to see as the disease progresses, you'll start to see the actual areas, those file cabinets start to degenerate as well. Plaques form, preventing information from running up and down those axons properly. If you're ever interested in scaring the living daylights out of yourself, you could look up something called a prion protein. And these are abnormal proteins that when they come in contact with other normal proteins, cause them to fold abnormally. Anybody ever hear of mad cow disease? That's what this is caused, or is the culprit of that. These prions, these strange little proteins. Scary as all get out. So if you want to look that up as a side.
See what it can do.
Yeah. And that's a brain degenerative disease where we start to basically eat the brain away. And then neurofibrillary tangles, when neurons actually start to kill off each other because of their tangled web, and brain can shrink. Parkinson's disease, another degenerative disease where we start to see degeneration in dopamine-releasing neurons that are going to help stimulate a lot of functions. When we think of Parkinson's disease, we think of a great deal of skeletal muscle involvement as well. So when we start depriving basal nuclei, those activating centers of dopamine, they become overactive and start to fire off neurons that cause muscle tremor, muscle contraction. And it's not as a controlled muscle contraction as we see in the normal individual. Exactly, and twitches and uncontrolled movements. Cause, again, researchers are constantly trying to figure out causes. Mitochondrial abnormalities or protein degradation pathways are high on the list for researchers as far as this disease goes. Huntington's disease, also a problem with allowing message to be sent through these pathways because of a buildup of protein. The protein is called huntingtin. And again, we see degeneration of pathways, especially in the basal nuclei, information trying to get to the cerebral cortex to access files that can't. So again, problems with controlling movement. So areas in that midbrain region that are responsible for coordinating movement are having troubles. Spinal cord, page 464 and 465. What is the spinal cord? It's a huge bundle of all sorts of stuff, basically taking information from peripheral nervous system, sending it to brain for processing, and then allowing motor tracts to leave the central nervous system to cause results. So basically what we find when we look at the spinal cord is groups of motor and sensory axons, as well as groups of nuclei that process information. This is where we find a lot of our interneurons or association neurons as well, hooking up information from sensory to motor. Where is it located? Down through the vertebrae, the spinal column. Begins at the foramen magnum, which is what? The big hole in your skull, at the base of your skull. And it doesn't go all the way down the spinal column. It actually ends around the first and second lumbar vertebrae. Now there's more wires down there. When I say wires, I mean nerve groups that fill up that space in the lumbar region. But those are coming from where? Yeah, all your lower extremities are feeding into the base of the spinal cord. So basically what we have, I described as wires feeding into the base of the spinal cord between L1 and L2. They actually look like, and they're called the phylum terminale and the cauda equina. They sort of look like a horse's tail when you look at them during dissection. So, yep, horse's tail. So we're going to provide two-way communication with the spinal column, spinal cord, and we're also have nuclei there for reflex centers, which we're going to talk about in this chapter. So remember I talked about finger on the hot stove. You're going to move that hand before you even realize you've touched the hot stove because of these reflex centers in the spinal cord. So, who's going to provide protection for the spinal cord? Bones. How many bones? 206? Not all 206. Do the math quick. 7, 12, 5, 7, 12, 5. 12 and 7 is 19, and 5 is 24. And then, of course, we have the sacrum and the coccyx, but they're not really providing protection for the spinal cord proper. They're providing protection for all of those nerve groups that feed into the spinal cord. We also have meninges that run down the spinal column. 3, what are they? Dura mater, pia mater, and then arachnoid in the middle. And then we have spaces where cerebrospinal fluid is going to flow. Fat is going to provide cushion in the spinal cord region. And then we also need a place to hold in all of the massive amounts of blood vessels that are going to help feed the neurons of the spinal cord as well. So we have cerebrospinal fluid in those spaces. We have the meninges, the dural and arachnoid membranes that extend all the way down to the sacrum. If I want to do a lumbar puncture, what's a lumbar puncture? Page 466, figure 12.27. A spinal tap. For what? What do I want? Who do I care about what's in spinal fluid? Yeah, you can check for a whole bunch of different things when you get a sample of cerebrospinal fluid. When I used to work in the chemistry department at the clinical lab, we'd get a sample of cerebrospinal fluid and we'd run it through our machines and we'd do a protein and a glucose test. Increased protein, there might be some organisms there that are protein-based. Decreased sugar, they might be eating that excess sugar. So we can find out a lot from a little sample of cerebrospinal fluid. That continuation of the meninges that goes all the way down creates the termination point of the spinal cord at the conus medullaris. And then we have a tap point that sort of continues down and holds everybody in so that the spinal cord doesn't bungee up and down inside the spinal column. That would be bad. Yes? Why would that be bad?
Because elastic bands break.
Well, think about it. We have all of these wires feeding into spaces between. Yeah. Now, if that spinal column doesn't stay in place and bungees up and down, it's going to do what to all of those things feeding in? Yeah, it can break them, it can pinch them, it can cause all sorts of problems. So we have something called the filum terminale that holds everything in place and anchors the spinal cord so it doesn't bungee up and down in the spinal column. And then we have denticulate ligaments that help to feed in some of the nerve groups and hold them in place as well. Those are extensions of the pia mater that we see holding everybody in place. And this is an example of a lumbar puncture. So we want to go well below where the typical spinal column ends. Are we all built the same? No. So do we want to go up at L1 or L2? Well, you said it ended there, so why don't we go L3? Well, what if yours is a little bit longer than mine? So where do we want to do our lumbar puncture? Yeah, as low as we can, so usually between L4 and L5. A needle is put into that area and spinal fluid can be extracted from there. So, spinal column consists of a whole bunch of wires feeding in. We have areas where there's a bunch of wires from these top extremities that create sort of a bunch more wires than maybe a little bit lower and create a larger area called the cervical enlargement in the spinal cord. We have cervical spinal nerves that feed in from either end. We have thoracic spinal nerves, and we have lumbar spinal nerves, all named for what? And those bones, yeah, you already know that. And what's the sacrum? Sacrum, when you looked at it in the lab. What the heck are all those holes? There you go. So those are foramen for nerves to feed into the spinal column. So the lumbar region has some extra wires that feed in and cause the lumbar enlargement. And then the conus medullaris is the end of the spinal cord proper. And then all of those wires feeding in from the lower extremities create this cauda equina, or horse's tail. And then the extension of the meninges is going to come down, tack everybody down, and create the filum terminale. Yes?
Yes.
So, how many spinal nerves do we have? 31 pairs. We have cervical and lumbosacral enlargements in the lower region and in the upper region because of what? Because of those extremities. We have more stuff feeding in, more wires. Cauda equina is a collection of nerves that are going to feed in from lower extremities. Now, ventral, dorsal, anterior, posterior. We have little infoldings of the spinal cord there as well. One's called the median fissure, anterior ventral, and one's called the median sulcus, posterior dorsal. Now remember, this is a pathway for information. It's like a train station. Information coming in, information going out. If you have ever gone to a train station, it would be very bad if we had train coming in and train going out on the same track. Yes? That would be a problem. It's also a problem with the central nervous system. So we're going to see that these very distinct areas delineate very distinct paths coming in and going out. And gray commissaires are connective masses of gray matter that are going to enclose the central canal. What was the central canal? The big hole right down the center where what's flowing up and down? Or down and out? Cerebrospinal fluid. So we see when we look through one of the vertebrae, what kind of vertebrae is this? It's a cervical vertebrae. How do you know that? Oh, smart ones of the bones. Because of the what? Transverse foramen. Very good. So this is how we stack these guys on top of each other and how nerve groups feed in between the intervertebral spaces. We can see different groupings as they come in, a vast blood supply. We see that there's several different regions. Now, this is where the book gets a little crazy, and this is where I am not going to get so crazy. They describe all of these different pathways, up and down, in the white matter regions of the spinal cord. I am not going to get that crazy, okay? Because otherwise we'll be on the nervous system for the whole semester. So we see that different white columns can be found in the dorsal and ventral regions of the spinal cord. And when a nerve feeds in, a spinal nerve, it splits. And it's going to send information down different tracks. This is the dorsal end. So what are we looking at here?
That's the back.
That's the back. And then we have the ventral root. Dorsal is back. Ventral is what? Front. You got it. The lateral is side. So these different railroad tracks feeding into the railroad station in any given nerve group are going to different places. So the dorsal horn is going to receive somatic and visceral sensory input. Information coming in is going to break off and go dorsal. The ventral horns is going to do what? Different tracks, different trains. What is that different track going to send from the central nervous system? Motor. Information out. I know it says right out. That's why somebody say something. So I know that you're awake. So lateral horns can be found in the thoracic region around that spinal, thoracic vertebrae and the muscles that we see all between the ribs. And we see some sympathetic neurons. What's a sympathetic neuron?
One that cries for the other one.
One that cries for the other one? No. I found out there was hermits for sale in the bookstore.
Right, it's not
Speed things up. Sympathetic neurons. Parasympathetic slows it down. Once I get my cookies, I'm good. Dorsal root, sensory input to cord and dorsal spinal ganglia, where we're going to see sort of a way station, or when you go to the train station, sometimes you have different locks, yes, that will switch information to different places. So think of the dorsal ganglia as that lock station that's going to help transmit information down the right track, going in and out of the spinal cord. So we see groups of cell bodies in these ganglia that are going to help with that information. Zones of spinal gray matter. Again, there's some processing that's going on very quickly, and a lot of that processing might involve reflexes, but might involve transmission of information. So we see somatosensory coming from what? Soma sensory. Information coming in from the body. Visceral sensory coming in from your internal organs. Visceral autonomic motor coming out, going to those organs, yes? And then somatic motor coming out and going to that stuff, yes? So see the difference? So we find that gray matter map in these little horns that are created in that central gray matter region in the spinal cord, mapped out as such. So what's SM? Somatic motor. Where is it? Motor. Where's motor? Outgoing. Where is it on the spinal cord? Ventral or dorsal? Ventral. See it there? Again, another motor region. Visceral motor is going to be found where? Ventral. So we can send message out. And you can see how it hooks up to those pathways. In between the two, what do you think we have? Yeah, we have interneurons and connections for things like reflexes. Me pulling my hand away from that stove really quick before message goes to my cerebral cortex. So in the dorsal horn, what are we going to find? No. Sensory. Visceral sensory and somatic sensory. And we see how those pathways are related in the spinal cord and the spinal nerves that feed into the spinal cord. Yes? So what do you have to remember? Ventral. Yeah, you're going to remember everything. But ventral, out. Dorsal, in. Yes? White matter tracts, all around that gray matter stuff. Those are the train tracks going up and down, carrying information for processing, whether it be processing in gray matter in the spinal cord or processing upstairs in the cerebral cortex. So we have myelinated and non-myelinated nerve fibers that are going to help connect all of this information from the peripheral nervous system. They run in three directions: up, down, and sideways. Yes? You got it. Ascending, sending sensory. So for me to do this, I have to send information from upstairs, down, and out. Yes? So hopefully that's self-explanatory. I don't need to say too much more on that one. We have three different major white columns on each side, left and right, controlling what? The left and right sides of your body. Dorsal posterior, lateral, and ventral anterior white columns. So, some of the major spinal tracts: relay, somatotropy, symmetry, and decussation. Decussation, excuse me. And those are different pathways from up, down, side, side, white matter train tracks. Don't get afraid. I'm not going to make you memorize it. But these are actually mapped out, some of the different pathways. In red, we have descending tracts. Remember, descending and ascending aren't going to be on the same track. It's not going to work. Yes? So we have problems with information that's being blocked somehow.
Oh, they're pretty close right there.
Yeah, they're pretty close, but they're not on the same track.
Well, they're split.
Yeah, they're close, but they're close is right. So you have to be very careful when we talk about damage to the spinal cord, and different drugs might cause these tracts to overlap each other and then cause a problem with relay of information. So we have ascending tracts going up. Remember, this is what? What end is that? Dorsal is sensory, yes? Motor is ventral. So we see the tracts outlined that way. All right, so ascending, what's that? Going up, sensory, consists of 3 neuron connections. First order neurons bringing information in from cutaneous receptors. What's cutaneous? Yeah, exactly. And proprioceptors. That inner ear stuff for balance. Branches, spinal cord or the medulla, and synapses with a second order neuron. The handoff guy, yes? Then the second order neuron, usually what we call an interneuron, is going to be associated with some sort of cell body in that dorsal horn of the spinal cord, processing the information, telling it where it needs to go, and axons extend to the thalamus or the cerebellum. That third order neuron, again, another interneuron in cell bodies in the thalamus region. So now we're up, we're past the spinal cord, and we're going up, and we're hanging out in midbrain regions. Now those midbrain regions have to do what? Yeah, they have to tell that information where to go, which file cabinet to access. So we extend axons into somatosensory cortex to then process the information. So there's no third order neurons in the cerebellum. Everybody gets extended or shipped off to that cerebral cortex. So 3 main ascending pathways. The dorsal column medial lemniscus pathways. You don't have to memorize this. The spinothalamic pathways and the spinocerebellar tracts. These are all train tracts going up the spinal cord for processing of sensory information in ascending pathways. I'm not going to get crazy about these guys. Recognize them as ascending pathways. And you don't have to memorize that much more. These guys are coming in, going up. Okay? When you look at this diagram, we see that pathway of information lined up for us on this diagram. So dorsal, column, medial, lemniscal pathways start with some sort of sensory receptor, tickle my feet. That information that then gets sent to through a spinal nerve to the spinal cord, where we're going to hook a what? In the diagram, we're going to hook a right, and we're going to go where? It's sensory information. Where do I want to go? I don't want to smash into motor information. We're going dorsal, so we're going to the dorsal horn. Then we're going to send that information up. Now, we might also send information out as we go up. And sometimes that information crosses over from the right to the left side of the spinal cord to get to its proper pathway. And that's what some of those interneurons will help us do in the spinal cord. So this is an ascending pathway. There's another pathway called the spinocerebellar pathway that sends information directly up the spinal cord to process in the cerebral cortex. So again, I don't want you to memorize these, but I want you... Yeah.
Because that doesn't have it doesn't go to the...
Correct.
It doesn't go to the...
That's going straight to the brain. So sometimes those reflex pathways will take the hardwired route and the other route as well. Spinothalamic pathways, again, description of pathways that get crossed over from one side to the other with a description of some of the first order and second order neurons. I'm not going to make you memorize that, so don't worry. Spinocerebellar tracts are going from the spinal cord to the cerebellum. And what do I need the cerebellum for? To help me what, my motor? Coordinate. To help me coordinate movements. So this is the train track information we'll take to help me coordinate movements. And again, they show us that pathway. I'm not going to make you memorize it, so don't. Just understand that those pathways are ascending and they're going to help with whatever they help with. Deliver efferent impulses from the brain to the spinal cord are descending pathways and tracts. These are motor. So now I need a result. Now I need to react to the stimulus. Two groups, direct pathway, the hardwired guy, or the indirect pathway. So we have information going out now in a descending pathway or tract. So motor pathways involve two neurons. Motor information coming from processing is going to be handed off by an upper motor neuron, and that's from the motor cortex. That's the, after I process, now I have to send motor out. Remember those different parts of the brain that we talked about? There was motor regions, sensory regions, and association regions. So now we're talking motor region. Lower motor neurons are going to pick up the message. That's going to be in the ventral horn of the spinal cord. Ventral is what? Front. So now we're coming down a different railroad track. Yes? And this is going to help innervate skeletal muscles. What's that mean? It's going to fire them off so they move. Direct pathway, impulses from pyramidal neurons in the precentral gyri pass through pyramidal or cortical spinal tracts. Again, I'm not going to make you memorize this, but this is a descending tract. This is motor. We see information being processed in the pyramidal cells in the cerebral cortex, in the region of the primary motor cortex, and then we send that information out and down the spinal cord. It can cross over so that it makes its destination properly, and the crossing over part of these information pathways are going to be achieved by those interneurons or association neurons, handing off information from one side to the other in the gray matter horns, dorsal and ventral, in the spinal cord.
So they're going to be pretty abundant in that area, right?
Correct. So there we see, and that's why gray matter, we see all those cell bodies. So then we have,
Just hit this straight. So gray matter is fast impulse.
No, gray matter is typically cell bodies of these neurons or unmyelinated fibers. Fast-moving stuff is going to be in the white matter.
That's what I thought.
Yep. But we need a handoff point. We need a processing point. So anytime we think about it this way, anytime we have to make a decision about where something is going, there's going to be some sort of cell body involved. Because the fibers are one-way go. So just like my dominoes, when I hit them down, Once they fire off, they're just going to go in one direction, and they're going to go fast. But there has to be some sort of decision maker as to who's going to fire off and when. So that's when we see gray matter and cell bodies. Does that make sense? So we need decision makers in the spinal cord too.
I get what's going on there, but
Decisions as to where the information has to go, or relay stations. You could think of it that way. Where I know I have to go in a certain track, but somebody's got to steer me in that direction. Indirect or multineuronal systems are complex and multisynaptic. A whole bunch of different synapses sending information to the pathways that need to be sent to whatever pathway we happen to be talking about. Brainstem, motor nuclei, and all motor pathways are going to follow this set of tracks out of the central nervous system. We have axial muscle maintaining balance and posture, and muscle controlling coarse limb movements. Head and neck and eye movements are some of the pathways that are directed in the indirect or multineuronal system. So some of the tracks out or motor. These are descending pathways. We need to maintain balance. How do you maintain balance? I kind of felt there was a place I could take a drink of coffee there, huh? In order for me to maintain balance, what has to happen to my body? Some sort of message has to be sent from the nervous system down those descending pathways in order for me to maintain balance. Certain muscles have to constantly do what? You have to send information to them so they can contract to maintain balance. So these are some of the pathways that are going to help me do that. Constantly send information, motor information, down a descending path that's going to help certain muscle groups contract on a regular basis to help me hold my head up straight while I'm conscious, for example. The cerebrospinal tracts are going to control flexor muscles. Flex. Yes? Superior colliculi and the tectospinal tracts are going to help with head movements. And again, these are some of the pathways. I'm not going to make you memorize that. Don't worry about it. Just understand that these are what? Descending pathways for motor information. And we see those tracks and where they run in the spinal cord. So, information coming in, information going out, very important part of the spinal cord. I disrupt that information in any way, and I can cause some serious problems with function. Exactly. Depending on where I breach this information coming in and going out is going to depend on or result in different results. Does that make sense? So, paralysis is loss of motor function. We can also lose sensory function. Remember, those spinal nerves split. So paresthesias is the loss of sensory, and paralysis is the loss of motor. So severe damage to the ventral horn, and what's the ventral horn for? Dorsal is what? Sensory. Ventral is motor. If I have severe damage to the motor end of things, the ventral horn, I'm going to have severe difficulties creating a response or motor response. So I might know what's going on, but I can't respond to it because I can't send that motor information to the muscles that need to move. Well, there's many demyelinating diseases that can result in this. A lot of the cerebral palsies have problems. Yeah. So you're going to have, when we affect the ventral horn, we're going to affect muscle movement. That can result in muscle atrophy, because if I don't use those muscles on a regular basis, and we're talking about thousands and thousands of muscle groups that are constantly being stimulated. If I don't stimulate them, they are going to start to atrophy. And that can, it's basically muscle death.
That's what happens when you break your arm and your arm's in a cast.
Yep.
When you take it out, it's real small compared to the other one. because
Correct.
the muscles atrophy.
Yeah, and then you have to
Exercises.
yeah,
to get
go
it.
to, or do some sort of physical therapy, whether it be through a physical therapist or on your own, to overstimulate those muscles in a way to build them back up and strengthen them. Spastic paralysis, damage to the upper motor neurons of the primary motor cortex. So spinal neurons remain intact, but we're talking about
The dorsal.
upper regions. Problems with voluntary control of muscles, and muscles often shorten permanently. Anybody ever work in a nursing home with a patient who has this type of paralysis? Try to unbend those people. It's impossible.
Oh. Oh,
Yeah. Transection, cross-sectioning of the spinal cord at any level. Again, wires coming in, wires going out. I cut the wires, no information. No information is coming in, no information is going out. Paraplegia results when I transect between T1 and L1. And then quadriplegia is when I transect in the cervical region. Yeah. Spinal shock, transient period of functional loss, caudal to lesion. So we can temporarily interrupt messages.
Also hemiplegia, right?
Mm-hmm.
Half the body, one side.
Correct. And that's, again, if I disrupt any of those tracts on either side, you can have partial paralysis or paralysis on one side of the body and paralysis, not paralysis on the other side of the body. I'm having trouble speaking today. Poliomyelitis. That's destruction of the ventral horn. Ventral is what? Motor. So problems with muscle atrophy because motor information can't get to muscles. Death may occur from paralysis depending on where the paralysis is.
That's short for polio, which is
Correct.
yeah.
So there's different extents of polio. It depends on what portion of the spinal tract was disrupted by the virus. If it's in the thoracic region, we might disrupt innervation of the diaphragm. And if we don't contract and relax the diaphragm, we don't what? We don't breathe. One of the things that came on site when polio was running rampant way back when in the old days is something called the iron lung that would help patients breathe because they couldn't innervate their own diaphragms. Different neuronal loss can result in different paralysis, thoracic region or down in the lumbar region with leg and lower extremity movement. Amyotrophic lateral sclerosis, or ALS, Lou Gehrig's disease. Terrible, terrible disease, but destruction of the ventral horn and motor neurons again. Losing the ability for motor function.
That right there happens extremely fast.
Yeah, and it happens very fast, and unfortunately we don't know how to stop it. Remember, and I speak to you or some of you who are going to go and take care of patients that might be suffering from these things. It's motor problems, not sensory.
Right, you know
They know exactly what's going on. They know exactly what you're saying, and they can process all that sensory information. It's the motor that can't function because of the death to those ventral horn tracts. Please always remember that. So even though your patient can't respond to you, they know exactly what you're saying and they know exactly what you're doing. So keep that in mind. Death typically occurs within 5 years. And again, depending on the health of the patient, it can go much faster than that for sure.
It was 30 years old, and it was less than a year.
Yeah.
It's gone.
So again, these diseases progress at very different rates depending on the patient's chemistry and stamina levels.
At the end stage, all they do is pretty much blink at you.
Yep.
That's about all they can do for motor.
But remember, all that sensory is there, which is very frustrating. Might be caused by environmental factors, genetic mutations involving RNA processing. Again, researchers are always trying to figure out what it is and if we can fix it. This drug, Reluzol, I think it's pronounced, interferes with glutamate signaling, and it's the only slowing the progression of the disease treatment that we have to date.
I have a question. You ever seen the movie Lorenzo's Oil?
I never actually saw Lorenzo's Oil.
Well, it's supposed to be, I think it's an oil, fat.
Yeah, which they're trying to treat,
And I
yeah.
don't know if you've seen or know what.
No, I should watch that. That would be good. Did you have a question?
Yeah, Stephen Hawking, doesn't he have ALS? But it stopped with him, correct?
Um,
More progressing?
It's slowly progressing in him.
And that's the only case that's ever happened.
He's the longest one that's been able to survive this.
20 years.
Yep. Yeah.
I just had a resident die that had it for
20 years. So yeah, it depends on chemistry, body chemistry. Ascending CNS dysfunctions, ascending going up. We do a bunch of reflex tests, which you're going to do in lab to test some of these ascending tracts with sensory stimuli. Okay. Exactly. Imaging techniques like CAT scan, magnetic resonance imaging, MRI, or positron emission tomography, PET scans, are going to help look at the integrity of these pathways in the brain and the spinal cord. Developmental aspects, we already kind of did this at the beginning, but for some reason this book put it at the end, and I didn't know that. So I already covered this stuff, but ectoderm, neural groove, neural folds, neural crest, neural tube, we've heard all of this before. On page 476, figure 12.34, we see these pathways starting in the embryonic spinal cord. So these are the beginning neurons that are going to form the gray matter regions, both dorsal and ventral horns, in the spinal cord during fetal and embryonic development. Developmental aspects, don't worry about it. Age brings some cognitive decline, but not significantly in healthy individuals until when? The 80s. So we have a decent span on this central nervous system and all the processing that goes on. Shrinkage of the brain accelerates with old age. Excessive use of nasty chemicals can result in pushing that 80 up significantly. So don't use nasty chemicals. You want to be able to process and function properly as long as you possibly can. And we already saw this at the beginning. That's our neural plate forming our neural groove. That's going to form our neural tube. That's going to start folding all over the place on itself to form the central nervous system. The homeostatic imbalances definition. So, on page 476, 477, just a definition of cerebral palsy, spina bifida. I didn't have them on there, did I? Okay. So again, we see them under on page 476 and 477, just a general definition.
I've seen that spinal fluid.
And that's it for chapter 12. Thank the good Lord, we're done with chapter 12. Woo! Phew. Yeah, now we have to have a quiz on chapter 12. Pull out your clickers. I'm going to do a couple questions. And I know you're going to hate my guts, but then I'm going to start chapter 13. All right. Which area helps plan movements? Wait a minute, let me start my engines. Oops. Movements, movements, movements. Two key words, plan and movements. 5, 4, 3, 2. Is that everybody? Oh, good. What's the answer? B. So you were on the right track when you thought motor movement, right? But we're going to plan the movement, pre-motor. We're going to initiate the movement in the primary motor. Yes? The phrases left brain and right brain indicate that the two cerebral hemispheres display a phenomenon called Five, 4, 3, 2, 1. What's the answer? The answer is C. Oh, good. Lateralization. Cerebral white matter in each hemisphere is greatest in amount for what? We're talking the cerebrum. Five. Oop. Oop. Answer's A. I didn't 5, 4, 3, 2, 1 you. Good. Good, good, good. I clicked too many times. And one more. Damaged basal nuclei, as in Parkinson's disease, results in Five, 4, 3, 2, 1. What's the answer? A. So these patients don't have a problem creating memories. These patients don't have a problem storing memories. They only have a problem what? Motor is affected. A lot of these diseases are like this. And it's so important for healthcare professionals to remember that when you're treating your patients. They might not be able to respond to you, but they understand everything that you're saying. Okay? So that's important to remember. Yes. All right. So, don't do it. All right, it's not even worth it.